Greetings, grade 12 learners. Welcome back to our Life Sciences Paper 2 revision. We are carrying on with the exam tips. Remember, in the previous lesson, I did say that you need to know your terminology because that is where you can easily get marks there. We usually have 1 up to 10 questions. So if you master your terminology, you'll be able to get all those 10 marks there. Now, this is just an example of a terminology question from one of our past papers, and it's looking at terms from different topics. The first one is talking to the position of a gene on a chromosome, which we know that it is a locus. Remember, spelling is very important. Once you put a T at the end of that locus, it becomes a locust, which is something that exists in life sciences. Therefore, we cannot say, no, shame, he knew that one. But if you say it's a locust, then you don't get the mark. The type of evolution that is characterized by long periods of little or no change, followed by short periods of rapid change, that is punctuated equilibrium. The natural shape of a DNA molecule is a double helix. Be very careful that you do not say it's a double-stranded helix. It is a double helix. And then the type of bond which is found between two amino acids, it's a peptide bond. It's not a polypeptide bond. A polypeptide is basically a chain of amino acids that are bonded together by a peptide bond. So don't make that mistake of saying it's a polypeptide bond or a peptide bond. Then the type of vision shared in primates that allows for depth perception, it's stereoscopic or binocular vision. The type of dominance, which results in an intermediate phenotype in a heterozygous condition, that is incomplete dominance. The key here, what is the key when we're talking about incomplete dominance? It's the intermediate phenotype. So if you have red and white, you get pink. As we said earlier, do not confuse this with co-dominance, where you will have both phenotypes being expressed in the organism, or you confuse it with complete dominance. Complete dominance is where you have one allele dominant over the other, and the allele that is recessive will be masked by the dominant allele. The fluid of the nucleus, where free nucleotides are found, is a nucleoplasm. A tangled mass of chromosomes located within a nucleus is a chromatin network. Please do not just write its chromatin, because it was saying a tangled mass of chromosomes. If we said that the network of chromosomes within the nucleus, then we would accept if you said chromatin. So you must understand why we wouldn't accept chromatin network, because we didn't include it. It is called the chromatin network, not just chromatin. Can we move? Then this is another example of a question in section A, the item or statements column questions, where you are given a statement in column 1 and then an item in column B. So here, what you need to know is that you need to pay attention to the instructions. The instructions say that if you must either write A only as your answer, if it only applies to A when you read there, or you must write B only. Don't leave the only. It's important that you write the only, or you must say both A and B. So please be sure that you follow the instructions. That is what is very important here. So some of the common errors that the learners are following is that they will write 2 letters, or they will write A/B. Once you write 2 letters when the answer is actually A, and you write A or B, then you don't get the mark. Or you must write it as I indicated, write it as in the instructions. Right. Let us move to check the multiple choice. Now this is a multiple choice. It's not a level A multiple choice because it involves a calculation. But here, if we read the question, it says a segment of DNA contains 31% of adenine in strand 1. Remember, DNA has got 2 strands. Now it says there's 31% of adenine in strand 1 and 12% of cytosine in strand 2. So let us take this as strand 1 and that one as strand 2. Right. Now it is telling us in strand 1 we have got 31% of that is A. Then it tells us that in strand 2 we have got 12% of cytosine. Then again it tells us that we have got 27% of guanine. Okay, now what do we know about DNA even before we go to the question? We know that the amount of adenine will always be equal to the amount of thymine. So if it says 31% adenine here, it means that we are also going to have 31% of thymine in strand 2. Here it says 12% of cytosine, meaning that we are going to have how many of guanines here? We are going to have 12% of guanine. I hope I'm right. And then it says 27% is guanine. So here we're going to have 27% of cytosine in strand 1. Now we need to know what is the question saying. It says in this segment of DNA, there will also be 31% of adenine in strand 2. Right. So it means that here in strand 1, we are short of the percentage of thymine because we know what is the percentage of adenine, we know what is the percentage of G and what is the percentage of C. Now all these percentages, they should add up to 100. So if it is 31 plus 12, that is going to give us 43. And 43 plus 27, that is going to give us 70. So which means that we are left with what from 100%? 100% minus 70, it gives us 30%. It gives us 30% of T. Oopsie. It gives us 30% of thymine. I hope you are following me. And then that means that here we are also going to have 30% of adenine. So let's look at the first statement. 31% of adenine in strand 2, is it correct? No, it's wrong. 12% of cytosine in strand 1, it is not right because we only have 27%. 31% of thymine in strand 2, we can see that that one is correct because we've got it there. And then 27% of cytosine in strand 2. We don't have 27% of cytosine, we have 27% of guanine. Therefore, our answer there would be C. So that is how you are supposed to work out that answer. Right, let us move into this one, which is looking at the interpretation of a of the DNA profile. It's a case of paternity whereby the family wanted to identify. Remember, we said we must always underline the key things. They wanted to identify the biological father of a boy. They know who is the mother, who is right here. The mother, we can see, is blood O. And they took the blood groups as well as the DNA profiles of the mother and the boy, and the two males were used to determine paternity. So how do we find out from a DNA profile if a father is really a father of the child? We first have to compare the DNA bands of the child to the DNA bands of the mother. So we eliminate those that match. So we can see at this point here, we can see that at this part here, the boy is matching with the mother. And we can also see here that they are matching. And we can also see that they are also matching there. So I think those are the only ones where the boy is matching with the mother. Then what you do next, you look at the remaining bands from the boy and look at the fathers that are under discussion or under the question of whether who is the father of this child. So you look at the boy and you can see that the boy is matching with male 2 over there. And then you go down, you can see that there is a match with male 1, and there's also a match with male 2. And then you can see when you go further, where else they are matching. So there is only one place. Okay, we can also see over there. No, it's not matching. We can see that it is matching also here at the bottom with both males. But you can see that 1, 2, 3, where else is this matching? There's 4, sorry. Okay. Now, how many bands we can see here? If you look at male 1, there's only 3 bands there that is matching with male 1, whereas there are 4 bands, or all the remaining bands of the remaining bands of the boy, they match with the bands of male 2. So you first eliminate the mother's bands that matches with the boy, and then you eliminate those that match with the father. So male 2 would be the one, and the explanation as I have explained. Then let us look at this one. How can we then determine the paternity now using the blood groups? It says, explain why paternity cannot be established using only blood groups. Now, from the DNA profile, we know that the mother is blood group O. So which means that their genotype is going to be II. Okay. Then the boy is let us look at the fathers first. Male 1 is AB, so which means that they're gonna be IA, IB. This is an example of codominance where you have got 2 alleles being expressed in the phenotype of the male. And then here male 2 is IB. They can be IB with a small I, or they can be IB. Okay. Sorry, there should be another I here. So it's I capital B, I capital B. Then now let's go to the boy. The boy is type B. So definitely one of the alleles is going to come from the mother. So the boy is going to get 1 I from the mother. Then 2 males, both of them, they do have the IB allele, which means that we can't rely on the blood groups to get the, because the males both could give that boy the IB allele. So the genotype of the boy is going to be IB small I. Then they get the small I from the mother, and then the capital IB, they get it from the father. So both males, because they have got this IB allele. So if we only use the blood groups, any of those males, because of that IB allele, they can be a possible father. So that is the explanation there for the blood groups. So it says male 1 could be the father of the boy, or male 2, because they both have the IB allele, and as I've showed you what their genotypes are, and then the child gets the small I from the mother, and therefore he would have the genotype IB small I. Then this is just another diagram which is a part of DNA code of life, and it is representing a stage during a synthesis of a protein. Once they talk about synthesis of a protein and a stage, you either going to think about transcription, and you are either going to think about translation. In transcription, you should see a DNA. It should be in the nucleus, but here you can see that we have got these amino acids. We have amino acids, and we know that the amino acids, they are carried by the tRNA molecule. So definitely the process that is represented here, where you can also see this organelle labeled number 3, which is the ribosome. So we are showing the process of translation here. I hope you can understand. Why is it translation? Because you are having the different tRNAs bringing the amino acids according to the codon. This would be the mRNA here, right? So this is the codon in the mRNA, and that would be the anticodon on the tRNA. So the anticodon of the tRNA is complementary to the codon on the mRNA. So let's quickly look at the questions, because I think I've already answered most of them. Identification of number 3 is the ribosome. The role of the molecule 1, which is tRNA, it brings a specific amino acid, or the required amino acid. You can't just say it brings an amino acid, because each tRNA molecule is carrying a specific amino acid, and it doesn't change, because that is determined by the anticodon that is on the tRNA. So the common mistake that our learners made when this question was asked in the exam is that they said that it picks up an amino acid instead of saying it picks up a required or a specific amino acid, right? And then 2.13 says give the sequence of nitrogenous bases at number 2, right? So this is number 2. This is the part that they are looking at. Here you can see that we have GGA as the codon on the mRNA. So if it is GGA, it means that at number 2 we are going to have CCU. It can't be a T here because this is a tRNA, and in RNA we don't have a thymine. Instead, it will be a uracil, right? Then it's talking about the change in the amino acid sequence, which definitely it means that there was a mutation where the alanine was replaced by tryptophan. They want us to explain the possible reason for this. It could have been a mutation on the DNA molecule that resulted in the change in the codon. Now let's look at the 2 amino acids that they are talking about. We have for alanine, it's GGG, and then it changed to GAA in tryptophan. So we have GGG as the anticodon, which changed to GAA. And then in the mRNA, you would have had CCC, which would have changed, which would have been CUU, that is for mRNA codons. And then in the DNA, it would have been GGG and GAA. So that would have been the sequence. So you are working backwards here. So the question is of a higher order. So the learners would have to think more when they are answering this question. So I am going to end there with some of these examples for DNA code of life. I hope you have enjoyed our lesson today, and please write your questions on the comment section. I will come back to you and answer all your questions. Thank you very much.